I started out as a citizen mycologist for many, many years, starting from about 1970 through about 1991. I was out collecting. And then I went back and got my doctorate, well, a couple degrees, because I needed a science degree before I got the doctorate. And I'm called a mushroom gone bad by the citizen mycologist, because after you go through all of this work, there's a sort of disjunct that occurs. And what drove you in, the passion that got you into this, it doesn't abate, but conversing with the people who haven't gone what you've gone through is difficult. Anyway, the title of the talk is Rooting Out Phaeocollybia, Lessons Learned and a Second Chance. And the second chance I'm getting courtesy of the United States government, and we will touch on that later. Now let's see if I can, yeah, good. Okay, the scope of my project is simply a standard regional monograph. Now there's a nice thing about Phaeocollybia. It's a honey of a genus to study. It's small. There's only about 100 species known thus far in the world, give or take 20. It's very finicky, so it is considered rather rare. And that means that although I've done the Phaeocollybia of Northwest North America, actually, with the addition of only about 6 more species, I will have done North America, because most of it is located on the Pacific Coast region. I went into this with the idea that I was going to learn everything about the genus, which is why it was a honey of a project. I mean, I didn't have to take a little segment of a genus, which I would have had to have done if I took on Inocybe. Brandon will speak to that later. First thing is ecology and distribution. Where does it occur? How do you find it? It's considered a rare fungus. Discovered after thousands of collections, it's not that rare if you know where to look. To completely go through the morphology, what is its form, how do you identify it, what are the parts that are important to know. The neat thing about this genus is it does have characters. It's not like agaricus. You can look at a dried collection. I can actually identify the dried collections to species fairly frequently. So it is amazing. To learn about the biology, discovered the thing is mycorrhizal. They didn't know that. It could be possibly parasitic. There's it's not been solved yet. Learned about how it developed. It was thought it was put in the gymnoti, which means no universal veil, by Fries. Guess what? Has a universal veil. Learned all sorts of wonderful things about it, because you have to dig. If you recall the previous slide, that's what it's known for. You see the little portion that shows above, like a little brown mushroom, but look at how much of it extends below the surface. It's a rooting mushroom. It's a very sweet little thing to go through. Then the first step was to do what I call Taxonomy 1, alpha taxonomy. You have your species concepts. They are morphological, morphospecies concept. You go through the morphology and you generate these hypotheses. And I was able to build, which I'll get in the next slide, on work that was already done. Went in thinking there would be 15 species. I've thus far added 10 species, which kind of drift up and down depending on what the molecules tell us. But I had the morphospecies concepts. You challenge those then with systematics 2, which is you go through the molecules and say, well, how valid are these? I lucked out. I was able to test them because I did all of the collecting. I knew when I got something that wasn't right. The Eppendorf tube lied, and I discovered, well, I had switched collections on the dryer, but because I went back and looked at the thing, I had taken the prime one, and it actually belonged to another species. And I was able to do this because I was doing all of it. Then you go into phylogenetics. That's where you introduce the sequencing, and you see, well, is this holding together? Most of the time it did. And then we're moving on to barcoding, and there's a reason for that. So as I called, Phaeocollybia is an enigmatic, uncommon genus, large enough to mesmerize and small enough to master. You can't ask for anything better than that. Okay, so you're back at the beginning. What do? You have to know your beast. Now you're going to generate a regional monograph. This was a doctoral project, right? But But the first thing you do is, as Ron said, you amass the literature. Who's studied this before? You go back to Fries. You come forward. Now, we were lucky because Alex Smith had monographed it, or contributions to a monograph, so he had identified 15 species. And Rufus, the green thing was there. So that was one monograph. The other one, contributions to a monograph, was Aegon Horax, 1966. 76, 77 thing. But there had been 2 works by Alex Smith, a number of assorted papers. Murrell named the first species from North America in 1906, but nothing much else. We just knew that you really jumped up and down when you found a Phaeocollybia. So you amass all of this literature, then what do? Well, I'm a reductionist, so I went the Excel spreadsheet route, and that was to formulate an exegesis, and essentially is the pileus. And so you divide it into things, and it's the color of the pileus. I spent something like 4 months on determining how you could make color a keyable character, and I discovered that there are 8 colors. And all of our descriptions have little shades of this and little shades of, doesn't help. Was it green or was it brown? That's all you really needed to know in a lot of instances. It's amazing how much time that you will spend on these little nuances and these descriptions that completely cause your eyes to glaze over, and they don't tell you anything. They tell you what the mushroom you gathered that day, it's variable, and it has all of these various shades depending on the moisture content of the mushroom. So you learn what you need to code by. So I had to decide, does one cell equal one character state? Sorry about that. Or do you have to complete going to diameters, what you have, but you do that for every specimen. In this case, it was for every collection, every type description, every what people thought they had. The next thing is it a diagnostic character or is it a developmental variation? So these are the things that you have to work out when you're trying to decide what the taxonomy is. Then you have the conflict between a hard drive and your brain. And that's one we're never going to really solve, because you really do need to color code or character code, generate it, see what it tells you. It will reveal amazing insights, but it still comes down to the fact, can you recognize a thing on a field? Can you zen it? And you just can do it. After you've been in a genus long enough, you know what it is. But how do you tell somebody else how they can tell what it is as well? All right, so the next stage is going to the field to generate all these specimens, or going to the herbaria in order to collect the things. So you collect, you borrow, you beseech from all of your friends. I mean, they would just run and they would they'd come in the mail, or they would say, I've got some Phaeocollybias for you, because everybody knew what they were. And then after you get all of these thousands of specimens or collections together, and actually you're looking at specimens here because they mix together. It's a sociable genus. And so somebody, a very good mycologist would put in, and they would put a name on a collection, and then you'd go through and you'd separate them into the various 2 to 3 species that would have been growing in exactly the same spot. So you then do your examination. This is the anatomical. You're still morphology here. You're just looking at it a little closer under the microscope. Okay, then I started in the Dark Ages. I mean, in 1991, the ITS was a big deal, and so that's what I used. But I did riff flipping, which means that you PCR the thing, you get it out. I developed a protocol where I could actually amplify the spores, immediately challenge it with a restriction digest, but these were only single digests, and many of you may not even know what I'm talking about with riff flips. But you get it all out, and then you have David Hibbett on your PhD committee who says, okay, and you need to generate a tree. And I look at him and I say, excuse me, this is riff flips we're talking about here. He says, nope, you can do it, I want you to map it. Well, he was right, you actually could. I'm not convinced it was science. There was an awful lot of art involved in generating that. And I used Phylip and I used Antisis, and they generated trees, but you change one character and different tree. Okay, then we've moved on into the recent time, and this is the advantage of being a contracted government worker. The government ends up generating sequence data for me. And I'll get onto that later. So now you end up with, after checking 360 specimens, I think we're up now to about 400 or 500 actually. We have a nice little set of sequence data from the ITS region. And I was so pleased when Conrad Schock came out, and yes, it's the ITS, because for me, it has been a godsend. It's a very, even in rift lips, you could tell species apart. It supported, for the most part, my morphological species concept. It was great. Problems. Well, one of the things I found was what we call DNA contamination in old types. Now, this was back in my rift lip days. And of course, I'm a student, right, in trying to do the DNA work myself. And one of the things is Egon Horak had said that the mushroom in the middle was identical to the mushroom on the right, so that he put Skatesii in synonymy with Californica. This was based on a micro exam. He had never seen it fresh, and he was doing a type study at that point. And there is the only way you can really tell them apart is through the pileopellis, but he wasn't looking at the cap structure. Well, then I went down in 1992 and was collected this mushroom that I called Californica, but then when I gave the rift lip test to it, it came out identical to what I thought was the DNA from the type of Californica, which was collected in 1937. And the rift lip, the gels were the same, no matter what enzyme I threw at it. Skatesii was completely and totally different. Well, that thrilled me. No, wait, I've got that backwards. Rufotubulina was completely separate from Californica in the DNA, which was identical to Skatesii. So David and I had a little discussion, because when I wrote my dissertation, he says, I don't understand. Why aren't you putting this in synonymy? I mean, Egon thought it was, I said, it's not. It's different. He says, but your DNA tells you differently. I said, it's wrong. So a couple of years ago, well, actually this year, Matt Gordon, who's working with the BLM, finally extracted successfully after the 4th try from Californica, and it does show that Rufotubulina is identical to Californica and quite distinct molecularly from Skatesii. So these are the things you have to know. You have to kind of follow your gut, but you can't be demented and naive and think, well, I know better than the sequence data do, but you have to keep checking those data until you are assured that, okay, you were wrong eventually. So what I essentially am saying is there's no more Rufotubulina. Anything that comes out in my publication as Rufotubulina is, in fact, Californica. Okay, other problems involve what I call a sea of sensus. Somebody gets an old monograph, they key it out, they put a name on the box, and they shove it in the herbarium. And it's the species concepts that are always the problem. You're just completely surrounded by these misapprehension. Another is the wrong holotype designation. I decided to select as a holotype following Eros Pramastos' suggestion, get something from the center of the thing, so I got something with a lot of specimens and everything else. Well, it was a synonym, and I have the species itself is real, but I have to go back and select a new type and get a new name, because you can't the type and the name go together. Hybridization was a problem in one of my collections, because I would get these allelic bands sometimes and sometimes not. Missing pseudorhizas. Now this is when you have the citizens, mycologists involved. They just love to, oh, I got your Phaeocollybia, and here's this little thing, they've left all of this in the ground. So that's a problem. Black gels, I think we've all been there. You know, you run the gel and you have a nice ladder, nothing else. Phylop and enthesis was a major problem, because I had to teach myself away from okay, 2 minutes, I'm going to move on. Moronic book vault managers, no money, and of course the major problem is no time. Okay, these are the funding agencies. The third item is the primary funding agent. But I did have reciprocity grants and things to get me through my dissertation thing. The 1992 Barren Redhead Rainforest Expedition was memorable, got me through into the field. It was wonderful. Joe Emirati, my supervisor, would bail me out periodically. Oh, don't worry about that. We'll cover the reagents you need. The graduate fellowship. Northwest Forest Plan, I wanted to touch more on it. I lost track of time, but basically that was a major, major boost because you have the whole government agencies going out, getting collections, giving them to me to identify, and I was able to look at these massive numbers of collections. BLM cost shares, and our man in Salem, my co-author on the publication, is in fact a BLM scientist. I don't know if I had the book, but did I leave it? Well, no, I had another one. Yeah, that one. We do have a publication. It's 220 pages, 580 color photographs. It was everything I wanted to do. I got to put it together. I got to have drawings. I did, everything else. This was underwritten by the Bureau of Land Management, and then they slapped a $71 price on it. Many people who were going to shell out $71 for a very small, rare. Well, it's a sad story coming up. There was also the 2000 Roy Halling Talamancan Oak Expedition, and he's unrequited. I owe him lots. And then, okay, those are my funding. Those are everybody I need to thank, the universe. This is I'm going to have to spend time on this. Okay, publications. You just saw this. There's been 9 scientific papers, new species named, popular articles, lots and lots and lots of government reports on my identifications. I had my dissertation was 391 pages, and I had this wonderful book. The galleys for this came the same day as I got the sequence data. Guess what? But even better, last October, Ron goes in to get the books because people were ordering them and discovers that the government, moronic tenders, taken a book that was supposed to be there for 50 years and had burned it. No copies are left. And I was pretty upset. And then I said, Ron, if we can wear sackcloth and ashes and stuff, maybe we can then use the sequence data in the thing. I mean, I knew it at the time the galleys came. I was able to cover our asses by saying, the keys, well, it really may go here and what have you. So that is the second chance. And as to our website, it isn't there yet. It's just a figment. I have no time. That's the major problem in order to do it. And so basically, expanding Phaeocollybia, even though I feel like I own this genus on the West Coast, I'd be happy to have anybody who wants to do the databasing, the web stuff, or just enter the data. I mean, it's all ready to go. I know what it is to get it on the web, and I just don't have the time. So, thanks.